this is an oral presentation on the design and validation of an expression vector. These are the outlines. I'll be talking about the function of my gene of interest, the recombinant DNA technology, the transformation, the quality check, and the expression of my target protein. So I am particularly interested in study of autism as it relates to oxytocin. Having known that the prevalence of autism in the past years in UK has risen tremendously. About 1 in 100 people are autistic in the UK from the last government prevalence survey, and research has shown that most autistic people have very low levels of oxytocin, which is a cyclic nanopeptide hormone that acts as a neurotransmitter in the brain. So in this project, I wish to express oxytocin and then use it for further downstream processes to check if oxytocin can reduce the visible symptoms of autism. The gene that codes for oxytocin is OXT gene, and I'll be talking about the recombinant DNA technology. For the insertion of my gene of interest into a vector, I used PUC19 cloning or expression vector. I use it because of its excellent features like its high copy number, the reporter gene, it has the origin of replication, lac promoter, multiple cloning site, detectable marker amongst others. I'll use DNA recombination strategy by first looking at and pointing out that my gene constructs that I had used primers that has the same restriction enzyme site. So in this case, I used R1 to cleave both my gene of interest and my vector so that the restriction enzyme can cut the gene at the site, leaving a sticky end. So the T4 DNA ligase was also used to ligate my gene of interest that I had cleaved into PUC19 vector. So talking about the transformation procedure explained in this flow chart. The ampicillin sensitive cells were used so that we can be sure that what we have growing in the ampicillin resistant media is the cell that had picked up the plasmid, and the treatment of the cells with calcium chloride is for promotion of the binding of plasmid DNA to the lipopolysaccharide of the bacterial cells. Then the ice cold cells, of course, were heat shocked at 42 degrees Celsius for 30 seconds to make E. coli cells permeable to the DNA molecule cells that took up the plasmid. The cells that took up the plasmid are also differentiated from those that do not take up the plasmid by growing on ampicillin supplemented media. So I'll do some quality checks to know if I had generated my correct vector by using 3 steps. I'll use the blue-white selection, which enable me to pick out all the cells that were resistant to ampicillin and then that had picked up my plasmid or my insert, or had picked up my plasmid with insert on the culture plate, and the one that has recombinant DNA without the plasmid. So the one with the insert will appear white, while the one without the insert will appear blue. Then I'll do colony here. I'll do colony PCR by designing my insert or my insert specific primer to detect the presence of my insert. I'll set up a standard PCR reaction using the supernatant of my lyse cell. Then I'll run my PCR product on a gel to analyze the product size. Then the positive clones will give a band, while the ones that are not positive will not give a band. Then the third one will be the Sanger sequencing, the mini prep of the clones. I'll subject it to Sanger sequencing. Then I'll confirm the sequence of the inserts, and then the sequence of the junctions between the plasmid and the DNA inserts. So I'll basically run Sanger sequencing. The expression of my target protein, I would be using the IPTG molecular biology reagent. I'll put it into my experimentally growing bacterial culture, and it will act as an inducer that will trigger the transcription of the lactoperon and induce protein expression. So the gene under the control of the lac operator, which is our gene of interest, the lac operator, therefore oxytocin will be expressed, and further downstream processes will be done for further purification of oxytocin peptide hormone. I think that will be the end of